So, test is on Wednesday,
Mm-hmm.
and I know that Dr. E makes his tests pretty much from the student learning outcomes
Okay.
that are on the PowerPoint. So we're gonna go over that, all of those, and then if you have any questions, we'll go over that.
Okay.
So we're gonna start at week 7. Yes, week 7, so and that's transport across the membrane. So critically evaluate how different types of membrane transport, passive, active, facilitated, regulate cellular homeostasis, and predict consequences of their dysfunction. So passive, so the 3 main transport types: passive, active, facilitated. Passive is no energy. So this is gonna happen from high concentration to low concentration.
Mm-hmm.
So it's just gonna move through diffusion, and that happens naturally. You don't need energy source. And active transport requires energy. So this needs ATP to pump a certain molecule against the natural gradient. An example of this would be a sodium-potassium pump, or one of the complexes in the ETC.
Mm-hmm.
And then facilitated. So this is when there is something helping a molecule move. So active transport is essentially always facilitated,
Mm-hmm.
because you're gonna always have a protein in the membrane that's using energy to pump a substance. But
Mhm.
passive transport can be facilitated, or it can be just regular passive transport.
Oh, okay.
For regular passive transport, so molecule would move directly through the phospholipid bilayer, and the only things that can move directly through it are small, non-polar, non-charged molecules. So, like for example, steroid hormones,
Mm-hmm.
they are non-polar and they are relatively small. They're like, so they're able to move through the phospholipid with no help. But for example, a glucose molecule, it is more polar, and it's pretty large. So glucose needs a transport. Like that's why we need insulin to activate the transport
Mhm.
channels to move inside of the cell. And then, for example, just another example, water. It's small.
Mm-hmm.
It is polar, but because it's so small, it can partly move through the phospholipid bilayer in a passive, non-facilitated way.
Mm-hmm.
But there's also something called aquaporins,
Mm-hmm.
which is a channel that facilitates water movement, and water is able to move at a much higher rate
Right.
through that. So it's like a channel. So pretty much if there's a channel, it's facilitated. If there's a channel, no energy required, it's facilitated passive. If there's a channel and energy is required, it's facilitated active. And if it's no channel and there's movement, it's passive. And then, so predict consequences of their dysfunction. So it's just, if it's not working, the molecule can't go to the other side. It can't do whatever it's meant to do. So like in a sodium-potassium pump, you're not able to establish that electrical gradient in like maybe in cystic fibrosis. You're not cystic fibrosis happens when the channel that allows chloride to move outside the cell in a passive facilitated way, it's not properly functioning. And chloride is a charged ion, so charged ions cannot like at all move through the phospholipid bilayer without help, because it gets repulsed really easily since it has a charge. So it needs a channel, and if the channel is not properly working, chloride can't get to the other side, and we get that like really thick mucus. Okay. So number 2, apply membrane transport principles to real-world clinical cases, explaining how transport defects contribute to diseases such as diabetes, cystic fibrosis, and epilepsy. So exactly what I was talking about. Epilepsy, that's when your neurons begin firing all crazy. So when your electrochemical gradient is incorrect due to sodium-potassium pump not being able to function properly, and then you get like weird neuronal activation. So like things like your muscles are moving without you wanting to move them. And then cystic fibrosis, so like I said, and then so the whole point of chloride moving out of the cell, it's the main problem in cystic fibrosis is not that chloride is not able to move out itself, but that since chloride cannot move out, water is not able to follow it.
Okay.
So water sort of follows water goes to the place where there's a higher concentration of solute in the solvent. So if you remember, like sometimes they draw a picture like this. So this is a semi-permeable membrane right here in the middle, kind of like a phospholipid bilayer, and then over here, like you have lots and lots and lots and lots of things dissolved, but over here you don't have too many things dissolved. And so over time, water from here is gonna move to this side, and then the concentration of water is gonna increase over here, and it's gonna decrease over here until there is an equal concentration of solute in the solvent. Does that make sense?
Yeah.
Yeah. This is osmosis. So chloride is moving out of the cell, and it's artificially creating this environment of high solvent, so water follows. But since it's not able to move out, water is not able to follow, so the mucus becomes dehydrated pretty much. So it becomes like really thick, harder to move, and it's harder to breathe because mucus lines are like air pipes. And then diabetes, if glucose is not able to get inside of the cell, it's obviously gonna stay in the blood because glucose is somewhat polar and it's pretty big. So if it's not able to get inside the cell, it stays in the blood. If it stays in the blood, it causes like a whole series of issues. Hey girl. So number 3 is design a research experiment to test the role of transport proteins or ion channels in a given physiological process, justifying choices based on scientific reasoning. To test the role of transport proteins or ion channels in a given physiological. So again, like designing an experiment, most likely it's gonna have to do something with these 3 conditions: diabetes, cystic fibrosis, and epilepsy. And you know, when you design an experiment, you have to make sure you have a control, you have to make sure you have an experimental, that you're keeping as many variables the same as possible except for what you're actually testing. And you have to have a clear hypothesis, and then at the end of your design experiment, you can say what you predict, if the hypothesis is confirmed or if it's rejected. And essentially, like if you are trying to test an ion channel or protein, so you have to set up two separate chambers that are separated by a semi-permeable membrane. So we're like familiar with that word, right? So it's like semi-permeable membrane. So this is just another word for phospholipid bilayer essentially, but it's a membrane that selectively lets some things through, some things don't go through. And then your control is most likely missing the transport protein, and your experimental is gonna have the transport protein. And then, like, my hypothesis would be that if I put this chloride ion channel on that membrane, then the chloride concentration is going to be equal between both sides because it's going to move through facilitated passive transport until the concentration of chloride is equal on both sides. But if there's no transport channel and I put chloride only on one side of my semi-permeable membrane, then it's going to stay on that side. So it's just something like that, and then you change which molecule you're going to be using depending on the question. So analyze, number 4, analyze how electrochemical gradients drive secondary active transport and formulate hypotheses on how their disruption could impact cellular function. Electrochemical gradient, Okay, so then there's secondary active transport, and so essentially this is mostly related to glucose. In some cases, so glucose can move inside of the cell through those channels that are activated by insulin binding. So when insulin binds, most cells are like start exhibiting channels that allow glucose to move in. But then there are some cells, like for example the brain, the heart, they always need glucose, they always need energy. So no matter how much glucose they have in the blood, and then for the glucose to get into the blood, it has to go from the intestine to the endothelial cells in the intestine, and then it goes into the blood. And that has happened with no insulin, and the body uses sodium glucose symporters to enable that. So it's So you have GLUT1, 2, and 3, and they're all insulin independent. So it's spelled like this. If you see this, then it's talking about the glucose transporters, and then glucose 1, 2, and 3 are insulin independent, so they do not need insulin. But they are using sodium to couple it together and move glucose across the membrane. So in the intestine, pretty much, so this right here is your intestine and food is going, right? And then there's a glucose molecule, glucose. And this is like the most, this is the epithelial cell. So this is the one that touches directly towards the lumen of the intestine. Glucose has to get into this cell first, and then it goes to the next and like through the body. So, this channel right here, there's a high sodium concentration and a high glucose concentration here as well, but then glucose is not able to move through, and sodium is not able to move through by itself either. So the body uses sodium and glucose to move them together to this side through this channel, and then you don't need insulin to move it. So.
And that's only when it's high concentration?
Well, glucose wouldn't be able to move through anyway, but if you had a little bit of glucose here too, it would still be able to move through. I'm just saying that mostly, after you have a meal, you're gonna have a high glucose concentration in your intestine, most likely, unless you are having like a no carb meal. And so now glucose is in this cell. This keeps happening, so there is a buildup of sodium and glucose in here. So, like, obviously in the body it happens all like at the same time, everything. But right here, if we just think, so this has been happening for like a minute, let's say. So, and like a lot of glucose has moved in here now. Glucose needs to move further now. But also there's a high sodium concentration here, so sodium is gonna stop wanting to move in here, right? Because the concentration is balancing out, so it won't be able to bring more glucose. So then glucose moves further through another channel by itself, just from the concentration gradient, because we went from glucose being here, and then we moved it inside of the cell, and now it's gonna go to the next cell through a channel, just because of the gradient. But we want this to be still happening, so over here there's a sodium potassium pump. It pumps sodium out, and it pumps potassium in. So this, what I'm trying to do is just trying to connect these two processes. So the sodium goes in here, and then it goes to this cell outside to make sure that the sodium concentration in this cell right here is low, and this keeps happening. So number 5 is synthesize knowledge of ion channel mechanics to explain neuronal excitability, action potential generation, and signal propagation in a nerve and muscle cells. So just like some basic terminology. So dendrites. This is the soma or the body of the neuron. Then this is like the hill or whatever. And then this is the axon itself. And this is the neuronal ends where the neurotransmitters are released into the synapse. So basic stuff. And then like these things right here, you know, you sometimes see on neurons, myelin sheaths, right? Yeah. So just, I think we maybe went over this already a couple times, but really quick. So neurotransmitter is released from presynaptic neuron into the synapse. It binds to the receptors on the postsynaptic neuron. So the postsynaptic neuron is, it has a gradient established already. And that's done through sodium-potassium pump by pumping 3 sodiums outside of the cell and 2 potassiums inside of the cell. Both sodium and potassium are positively charged, but because there's 3 potassiums leaving and there's 3 sodiums leaving, sodium leaves, and potassium enters, there's a difference in outside is more positive than the inside, so there is a difference in charge. So that's established. Then after the neurotransmitter binds, the channels begin to open. So pump is used to establish the chemical gradient. So we have a gradient, and then a channel is opened, and if this is the membrane of a neuron, this is inside, this is outside. So there's lots of sodiums in here, and lots of potassiums here. Channels open, potassium flows outside, sodium flows inside. And like, it's like more physics how the actual electrical thing happens, but just know that this induces a change across the membrane. And then if you really want to impress Dr. E, maybe this is part of your written response, then so when the neurotransmitter binds, it binds to, it opens ligand gated channels. So basically these channels open when something binds to a receptor. That's the first, like the initial thing. And then all the other channels, they open just because of that initial change in the charge. So like after sodium moves in and potassium moves out, the charge inside changes. It's an arbitrary change, like it's not actual, but it changes pretty much from negative to positive, and then the channels, like over here along the axon, like there's millions of these, and then so the charge becomes positive, positive, positive. This one opens, charge is positive here. This one opens, charge moves. Does that make sense?
So it's negative on the outside? Right?
Kind of, yeah, pretty much. Let's just say that. When the charge is going, it's negative on the outside. When neuron is resting, so neuron is not firing, it's less positive inside. So it is negative. We're gonna call it negative. But then when this happens, I just wanna like I'm trying to say like how the charge actually moves. So if we're gonna zoom in right here, okay, we're gonna zoom in right here. So this one right here was open because neurotransmitter bound to it, and then this right here induced this negative became positive, like in its area. It's only like around it in the very small amount. But this channel right here is really close to this one. So this one sees like, oh, the area around me changed from negative to positive, so I'm gonna do the same. And then this one sees that, oh, now this one also had its area change. Now this one also had a share. And it happens like in steps until it reaches all the way over here. And over here, that change in charge actually releases neurotransmitters instead of just opening another channel, and it goes to the next neuron. Then if this is messed up, you can have seizures, you can have muscle weakness, you can have maybe like deterioration of neurons. And then also so neurons are not able to regulate well, we are able to regulate intensity, but neurons are all or nothing. So it's either fires or it doesn't fire. It's like binary, if that makes sense. It's like either 0 or it's a 1. But neurons regulate intensity through frequency. So if there is gonna be 100 of firings every second, then you're gonna have like a intense pain or whatever. But if there's like 20 firings of a neuron per second, then you're feeling less pain. Alright, so now it's the next week. So number 1 is predict the impact of pharmacological or genetic modifications to ion channels on excitable tissues such as in neurological disorders and cardiac arrhythmias. Like, pretty much you're going to be using information from this to answer a question about like some sort of disorder. So like if it's muscle weakness, maybe one of these channels is messed up, and then you're gonna be like using CRISPR or whatever to change the DNA, right? Or maybe you're gonna use like some drug to maybe actually inhibit channels, so like the heart can beat in a more steady pattern. So if you have cardiac arrhythmias, most likely this happens like too fast in a way, and then you need a drug to actually like slow it down. But then if you have muscle weakness, this is not happening fast enough. So you need to maybe fix these channels through genetic modification to make it move things better. So number 2, interpret experimental data related to membrane potential, ion flux, and synaptic transmission. Draw logical conclusions from trends and anomalies. So we have a good understanding of how neurons work. Action potential. That should be good. Number 3 is propose targeted therapeutic strategies for treating membrane transport related diseases, considering both mechanistic and clinical perspective. Genetic modification or like medicine, maybe like some sort of physical therapy depending on what it is. But you really can't go wrong with CRISPR-Cas9 in most patients. Compare and contrast the molecular mechanisms of voltage-gated, ligand-gated, and mechanically gated ion channels, linking the structure to function in various physiological systems. So, it's in the name. Voltage-gated, it opens due to a change in voltage, like these right here we're talking about. Ligand-gated, so ligand is just a fancy word. It describes anything that binds to a receptor. So a ligand is something that binds to another receptor that's specific for that thing. So like insulin is a ligand, steroids are a ligand, like there's thousands of ligands, right? And neurotransmitters are also ligands because they have specific receptors that bind specific neurotransmitters. So, and in a neuron, it would be ligand gated and then voltage gated. And then mechanically gated ion channels. An example, it's like it opens literally when you press on it. So they detect stimulus from actual like movement. I can't think of a good example right now, but maybe like in the ear, you know, like the
Like the hairline of the ears? The hairlines of the ears?
yeah, but like the inside though, like you know, like where there's the, like what is it called? The cochlea, I want to say. Huh? You know the thing like that looks like a snail inside of the yeah. So you know there's liquid moves over there, and then there's hairs, and then like the movement of that with those hairs opens the channel. So like just it's a mechanical thing. And then number 5 is construct a mechanistic model explaining how membrane transport and electrical signaling integrate to maintain systemic functions such as muscle contraction, neurotransmission, and fluid balance. That's just putting all of this together. Okay, next lecture. Week 7 and week 9 are the same. Okay. Analyze how energy is extracted from glucose and fatty acids by tracing the molecular flow through glycolysis, the TCA cycle, and oxidative phosphorylation. So, energy creation happens in 3 steps. Number 1 is going to be glycolysis. Glycolysis happens in the cytosol. Glycolysis results in 2 molecules of pyruvate from 1 molecule of glucose. You use 2 ATP to start it, you generate 4 ATP, which comes out to 2 ATP net generation and 2 NADHs. So every pyruvate makes 1 NADH, so you end up with 2. Then pyruvate is changed to acetyl-CoA. Then acetyl-CoA enters the TCA cycle. The TCA cycle makes 3 NADHs, 1 GTP. So in glycolysis, you're making pyruvate from glucose. The net result is going to be minus 2 ATP plus 4 ATP plus 2 NADH. Then pyruvate goes to acetyl-CoA and it goes to TCA cycle. You don't need to know all the steps of the TCA, but maybe just remember that acetyl-CoA, right here, acetyl-CoA plus oxaloacetate, it comes out to citrate, and this is the first step. Once this is made, once citrate is made, you go through one full cycle. So like as soon as this is made, the things are going to happen until citrate comes back to oxaloacetate, and oxaloacetate is going to be waiting over there for the next acetyl-CoA. So it's a circle, right? And then this is made, so it has to finish it. Like as long as it does this first step, it goes all around. Then TCA, this whole cycle makes 3 NADHs, 1 FADH2, and 1 GTP. It's the same as ATP, so we're going to 1 ATP. Then if there's going to be a question asking you about describing the pathway from glucose until the end, you are going to be starting with 1 molecule of glucose, but from this you end up with 2 pyruvates, and each pyruvate by itself becomes a separate acetyl-CoA, and each separate acetyl-CoA can send the cycle through one whole loop. So essentially 1 glucose sends 2 TCA cycles. Does that make sense? And so from TCA happens inside of the mitochondria, in the matrix of the mitochondria. So on the inside. So glycolysis in the cytoplasm, inside of the cell. TCA, we are already inside of the mitochondria. And we're making this NADH and FADH2. GTP it's used directly like that. NADH and FADH2 go to the electron transport chain where they move hydrogen ions into the intermembranous space because they're using like facilitated active transfer. So they're dropping electrons off, and that dropping electrons off provides energy for hydrogens to move against their concentration gradient and create a buildup in the intermembranous space. We know what I'm talking about, right? And do you remember this? This is important. Like the whole ETC electrochemical gradient of hydrogens. Okay, so really quick, you'll remember right now. So it's this is the mitochondria, but this is inside. So this is the outer membrane. This is the inner membrane. This is the intermembranous space, like between the two, and this is the matrix. And so NADH comes over here, it drops off an electron, and that just pumps hydrogens in here. So it moves all of them over here. So we are creating a fake gradient. So now, if there was a channel that would allow these to move backwards, they would with no energy. Because high to low is easy. Right here, we're going low to high, we need energy. But then moving backwards, we're going high to low, so we don't need energy. And then they are moving through a channel called ATP synthase. And this is a channel that allows it, because it's charged, it can't move easily, just like chlorine. Charged, it needs a channel. So this channel, hydrogen moves through, and this movement generates that ATP. So kind of like, we are doing all of that specifically to create this gradient, specifically so we have hydrogens moving. So all of this is just like building up for this one final step.
Okay.
And every NADH makes approximately 2.5 ATP. Every FADH2 makes approximately 1.5 ATP. You can't make half an ATP, you can't, but this is just because sometimes this process is inefficient. In reality, like when we're talking about it, NADH comes, it pumps hydrogen, but sometimes it pumps it, sometimes it doesn't pump it. So it makes between 2 to 3 ATP and it makes 1 to 2 ATP. You can't make half an ATP, but it's just average math, you know. So number 2 is explain the regulatory logic behind reciprocal control of glycolysis and gluconeogenesis, including hormonal and allosteric control points. So, like hormonal control, it's when you are changing gene expression inside of the cell, most likely. So, for example, you're gonna have more enzymes that participate in the TCA, so it happens faster, maybe in the glycolysis, or maybe there's more channels for glucose to move in, so you're able to upregulate it. So hormones most likely is gonna affect gene expression, and it can be either upregulated or downregulated. And then allosteric control points, so that refers to enzymes specifically, and allosteric control is when something binds to the enzyme at its non-active site. So the active site is where the main reaction happens. And then you have allosteric control point. So it's not a competitive inhibitor. That's the main point. Allosteric means that there's a separate binding site. Competitive inhibitor would bind to the active site, and it would just prevent a reaction from happening, like if somebody is standing in the door, you can't move through. But if somebody locks the door, you can't move through because of something else, right? So like allosteric is like the key. So it just binds right here, and the enzyme goes through a change, and it's not able to bind to like a target molecule anymore. Or maybe the other way, you bind to it, and then the enzyme is activated. And so like feedback inhibition is going to happen through allosteric regulation. For example, in the TCA cycle, like if there is a large buildup of NADHs, like the product, then some enzymes are going to begin to slow down because they're going to see that there's a lot of NADH in the cytosol, in the water inside of the mitochondria, and then that's going to prevent them from doing this, because why would you want to keep doing this if you have a lot of NADHs already? So that's a prevention of like kind of us wasting energy. So predict cellular responses to metabolic stress such as hypoxia, fasting, or mitochondrial dysfunction using pathway-specific reasoning. So hypoxia is low oxygen. Fasting is low glucose essentially, or mitochondrial dysfunction. So right here, so oxygen is the final electrons acceptor of the ETC. So without oxygen, you're gonna be doing, you won't be able to get this payoff right here. So essentially in hypoxia, you're gonna be stuck in glycolysis, and like in yeast cells, that's how fermentation happens. In us, just remember, in hypoxia, you mostly rely on glycolysis, okay? Because over here, you're making 2 ATP, but you do not need oxygen whatsoever. So if you don't have oxygen, your body is trying to get whatever ATP it can, and this is the best route for it. So it just keeps going glycolysis until you run out of glucose. Then in fasting, you're pretty much so. So glucose neogenesis generation. So it's generation of glucose from non-sugar molecules. So if you have low glucose, your lipids are gonna start undergoing gluconeogenesis. And then once you run out of lipids, your proteins are gonna be degraded to undergo gluconeogenesis. So that would be a possible response to fasting. And then a response to mitochondrial dysfunction. Mitochondrial dysfunction usually is gonna end up with an increased amount of reactive oxygen species. So ROS inside of the cell is gonna start leaking out. If these start leaking out, the cell is gonna go through apoptosis. It's gonna kill itself. There can also be DNA damage, or maybe there's gonna be an upregulation of antioxidants, like we've heard that word before, right? Antioxidants. So antioxidants combat reactive oxygen species. So like this one, when combined with this, it makes it neutral. I think it just makes it into water, I'm pretty sure. And so these can be made by the body, or this is also, I'm pretty sure, vitamin A and E, and also there's like other molecules that can act as these. So maybe mitochondrial dysfunction, there's gonna be an increase in the reactive oxygen species, which might result in cellular death or production of antioxidants. And number 5, design a conceptual experimental approach to test how altering a metabolic enzyme would affect ATP yield and metabolite flow. So again, if you have an experiment, you're gonna have control, you're gonna have experimental, you're gonna have a hypothesis, and you're gonna most likely have the control, like not have the enzyme, and then have the experimental have the enzyme, and that you're gonna measure the concentration of certain ligand molecule before and after, and then compare and reject or confirm the hypothesis. Okay, so I think he did not go over metabolism, so honestly, I think that's for the most part what you need to know for the test. So it's not too much. Any specific question? I'm gonna go over the pictures really quick.
On my note, where does the fat enter the TCA cycle?
So, to be honest with you, I don't know what he's looking for, because fat can enter from multiple places in the TCA cycle. It can, most often, if I remember correctly, it enters at the acetyl-CoA. So fat just goes through a different type of pathway. Yeah. So, like, fat is gonna be a glycerol, and then it's gonna be like this carbons, right? So it's like fatty acids, So it's lots and lots of carbons. And then acetyl-CoA, it's like 2 carbons plus a group. So essentially, like when you start to being hungry, when you're hungry, like you break off 2 carbons at a time, and you make it into acetyl-CoA. So you like pretty much you skip glycolysis and pyruvate, and you go direct into TCA. This is why, like, to burn fat, you kind of need to be doing like aerobic exercise, meaning you need to involve your ETC and TCA, because glycolysis would be anaerobic, hypoxic, low oxygen, but then so you're not able to burn fat in glycolysis. Glycolysis is strictly for glucose. Fat enters at TCA, so at the acetyl-CoA, and for that to be able to function, it needs a constant supply of oxygen. So just if that helps with like connecting it. So channels, just again, so we're clear, channels are just gates, they'll open or close, and transporters, they use energy to transport. I feel like he is gonna have a question saying which one of these glucose transporters are glucose dependent and glucose independent. So it's 1, 2, 3 are independent, and 4 and 5 are dependent. Oh no. Only 4 is insulin dependent. Only 4 is insulin dependent. So just remember that. 4 is insulin dependent, and then if you have to talk about it like in some sort of length, you know that like brain is a very like important organs, like the brain, the heart, the liver, they need a constant supply of glucose because they need to be functioning at optimal capacity always. So they cannot rely on presence of insulin, because insulin is only present like for some time after a meal. Then action potential, we're clear that refers to like the potential for the neuron to send a signal. So action potential is like the actual signal that travels across the neuron. And also, I forgot to mention this. I did say that it's like all or nothing, and then there's a threshold, right? So if one neurotransmitter binds, if like literally just one binds and opens the ligand-gated channels, it's gonna open, but it won't change charge enough for like the thing to happen. It needs to, let's say, it's not the correct number, but like the charge inside needs to become like, it needs to change by 20 millivolts. If it changes by 19.9 millivolts, nothing is gonna happen. You're gonna still get that 0. But then if it changes by 20 millivolts, it's gonna be a full fire. So it's all or nothing. Oh, and at the end, again, this is specifics. I don't know if he's gonna ask it, but when you get towards the end of the axon, there's voltage gated channels. They open kind of like in muscle. Calcium is released into the cell, and that release of calcium is what signals for the neurons to release neurotransmitters. Yeah, I don't know. I think that's it. I feel like maybe I'm forgetting something. Nancy, how did you do on the last test?
Let's just say that's it.
Better than on the first?
No, I think I did better on the first one. I was hoping to do better in the second one, but it got me more on the essay question.
Do
That's
you
where I was like,
do you start with it or do you end with it?